This is the first of the Unit 4 Biology revision sessions, and I am going to be covering the content required for the sampling techniques part of this module. So first of all, we're going to start with some keywords and definitions because, as you will be aware, in biology, keywords are really important. So if you take the time at this moment to pause this video, you can actually have a go at matching up the keyword and definition yourself, see if you can remember them, see if you've revised well enough. So if we go through these answers then, we can see, if we take some of the easier ones first, things like a relationship. So the relationship is an interaction between organisms. Okay, nice and easy. Sampling, again, we're looking at the counting of a small number of the total population of organisms. When we look at a community, we're looking at different populations living together in one area. The distribution is the location of organisms of one particular type of species, and population is the number of organisms of a particular species in an area. If these are of any difficulty to you, I think it's time you started revising again on this area. So we can sample different populations in different habitats to have a look at some of the different factors that might be affecting their distribution. Things like light and water and shelter and things like that. Now, the different methods that we can use include nets, pouters, pitfall traps, and quadrats. You may have come across quadrats. You may have come across some of these during your science lessons previously. Now, these are quadrats. They're usually a metal grid that you can put on the floor to measure the population of things like plants and small insects such as slugs and snails, as they don't move very quickly, so they're not going to move out of that area. These quadrats tend to be around 1 metre by a metre, so a metre squared. Now obviously, if you're trying to measure the population in a great big field like this, you're not going to be able to get an accurate, a definite number for the population, so what you're going to achieve is an estimate. So it's unlikely that you're going to get every single organism. If you're doing plants, you're not going to get every single daisy or dandelion that's in a field. But the more samples that you carry out in that area, the more accurate that your sample is going to be. Now, if you've got an area of field, for example, that is 50 metres squared, and you have a quadrat, your grid, that is 1 metre squared, you can use this to work out the population of a plant such as a daisy. So if your quadrat was placed in this area, you can safely count that there are 3 daisies in your quadrat. Now, the way that we can work out the population then is that we can take the 3 in your 1 metre squared and we can multiply it into the 50 metre square, which is the whole area of the field. Now, so that would be 3 times 50, because you've got 50 times this 1 metre squared quadrat in your area of field, and we will get 150 daisy plants. Now, this whole process to be random, because if, for example, we've got a little patch of daisy plants here, if someone was to decide that they was going to put their quadrat right over that daisy patch, then, for example, we might have 15 daisies in this area. If we then use that one quadrat to work out how many there are in the entire field, we would have to do 15 times 50, and we would get a number nowhere near to this. So you have to make sure that this is random so that it's a representative sample that you are collecting. And obviously, to round this off, if we just took one sample with one quadrat, with these two, we got two totally different numbers, really far out away from each other. This is why we need to do more than one. So the more samples we do in that one area, the more representative, the more valid it's going to be. Now, sweep nets are another way to be able to take a sample of organisms. Now, it tends to be used for flying insects such as butterflies and moths. We've got the apparatus here, which is a sweep net, which is wafted around long grass, trees, things like that. The insects will collect inside the net, and we will be able to count the number of different organisms found within that net. Now, this is a piece of equipment called a pooter. Now, as you can see, we've got this item here, which is usually a jar or a test tube as shown in the picture, and we've got a suction part where the person using it can apply suction to this tube, and then this part of the tube here can be put over the insect that we're wanting to catch, and the suction would suck the insect up through the tube and into the container here for observation and counting. Now, to ensure that we get no insects in our mouths, as no one ever wants that to happen, there is usually like a fine mesh over the entrance to this suction tube, so that if someone does apply a little bit too much suction, the insect won't get pulled up into the mouth. Here we've got a really simple piece of equipment for sampling. We've got the soil here with a small hole dug into it. Now, what is usually put in there is some sort of small plastic container, a cup, something like that. It's usually used to catch small invertebrates such as beetles, spiders, things like that. As we only want to collect these small invertebrates, we've got a roof on here. Now, that enables only small insects to come along under this roof and drop into the cup. We've got a funnel here to prevent anything getting back out and anything too big getting in. However, not all pitfall traps will have this feature. Some will just be an open yogurt pot, for example. So it's really important to check this piece of apparatus regularly to ensure that we've not had any predators eating the insects or any have not escaped. So the next sampling technique that we're going to look at is called the capture-recapture technique. The way that this works is that we imagine that this is a population of snails. We can't see them all. Imagine that there's some hidden grass and in trees and things like that. We go around finding in a particular area all the snails that we possibly can. We take them in and we mark the ones that we find. Here I've shown that with the blue dot. Now, what you would then do is that you would release them back into the same area that you captured them in the first place. When the snails are given a chance to reintegrate in with the population, you come back and take a second sample of the population. Now, the next time you come along to take this sample, it might be that you don't get the exact same individual as you got before. If the second time I've managed to capture this one, this one, this one, this one, and this one, you can see that we have got different set of organisms. Yep, 3 are the same, we've got 2 that are different. You would then count the number of marked individuals that you have recaptured. So here it's really obvious that we have got 3 individuals that were marked when they were recaptured. Now, the population size can then be estimated using this equation here. So we've got the number in the first sample times the number in the second sample divided by the number in the second sample previously marked. So the number in the first sample, we took 5 individuals. The second sample, we had 5. In the second sample, we had 3 that were previously marked. That will give us 5 times 5, which is 25, divided by 3, which gives us 8.5. 3 occurring. Now if we look back at our snails from earlier, our population of snails, when using this particular method for sampling, there are particular assumptions that you've got to make. You've got to assume that there's been no death. So all of them have survived between the first sample that you captured and the second sample that you captured. You've got to assume that none of the individuals have immigrated or emigrated, so none of them have left the population, left the area, and no new ones have entered. Okay, so we've got to assume that this one we captured here is not a new snail that has entered from a different area. We've got to also assume that the sampling methods that we use between the first sample and the second sample are identical, and that the marking of the snails has not affected the survival rate. Okay, so that this one here hasn't died because the marking has affected it, hasn't killed it, or this one here that we circled earlier has not been more visible to predators, so it's been eaten.